was midterm 2 basically chapter or lecture 7 through 12?
It is lecture 7. What did we just do? Was that lecture 13?
13 was gene interactions.
Yeah, so not that.
Okay.
I think it's just 7 through 12. Alright, so what do you guys want to know? The two of you.
Like any topics you want to go over, or?
This is your review session. I mean, I can't go over like 3 weeks of material in 1 hour, so you'll have to choose what's important for you.
Okay.
Student number 3.
Hey.
Welcome to the biggest review session of all time.
Do you have any questions?
Did you go over, let's say, transposons?
Anything particular about them?
Not really, just general view of them.
Sure. I mean, that was like a lot of the lecture, so if you can narrow it down a little bit, that would
narrow it down a little bit.
Otherwise we could talk about it for a whole hour. So if you, again, it'll help me if you guys have specific questions, concepts that you're not quite understanding, or problems that you have difficulty with. This is going to be the most boring video. So.
People are coming.
Hey, number 4.
Do you have a question?
Yeah.
Okay, cool.
Chapter 11, problem 22. That was a huge problem.
Okay, let's take a look at it.
And honestly, I got tired of writing the entire thing out, so.
Chapter 11, number 22.
Number 22
The trip opera.
is the tryptophan opera.
Okay. So, all right. So how would you start it?
Reading the question.
So the first part, they're asking you to basically draw a diagram of the trip operon, right? With these different elements. We've got a promoter. Next we have operator, sorry. And the first structural gene. Okay. So indicate where on the DNA the requested protein binds when it is bound to tryptophan. So we have 3 boxes here. Put an arrow in this one. All right. So where do you want your promoter to be?
The very first one.
Okay, good. And so what's going to happen in the promoter? What is that region good for?
Starting the transcription, but it's not actually going to be transcribed over.
So the promoter itself won't be transcribed.
Yeah.
But what protein is binding to the promoter? Hmm? So you say it's going to start transcription. So how do you?
Polymerase.
Okay, what kind of polymerase?
DNA polymerase. RNA polymerase.
RNA polymerase, right? So if it was DNA polymerase, that's not going to do us much good in terms of transcription. So RNA polymerase is going to be binding to the promoter. So what do you think the next part is?
Operator?
Operator, okay. And what are operators? What do they do?
Binding site for the repressor.
Good, okay. And so in this case, what's the name of our repressor?
Tryptophan.
Trip repressor? Trip up.
Trip A?
Trip-R.
TRPA, I think.
I'm sorry?
TRPA.
So
Like the
trip
official
A
name.
is the gene that's going to be transcribed,
Okay.
right?
Trip-A.
So this is the gene where we're going to initiate transcription and end up making this protein that's involved in tryptophan biosynthesis. Okay, and so this trip repressor here is going to bind the operator under what condition?
When tryptophan is present.
Good. Yeah, good. Just making doubt yourself.
Yes.
When tryptophan is present. All right, so we've got some tryptophan boundary. All right. So what does that mean? So if the trip repressor binds to the operator when tryptophan is present, what effect is that going to have on transcription?
No transcriptions going to occur?
That's right. So it's going to block transcription from occurring. I'm not going to have that going on.
So it's like.
So that makes sense. So in conditions where tryptophan is low, we want transcription to occur.
Mhm.
In conditions where tryptophan is high, we don't want to create the enzymes that will make more tryptophan because we don't need it.
Like feedback inhibition.
Okay.
Like feedback.
That's right. It's feedback.
Mhm.
It's a feedback loop. All right. So indicate where on the DNA the pressure protein binds on tryptophan. All right. I think we've done that.
Mhm.
Great. And essentially, this is serving as a physical block, so that you cannot have RNA polymerase proceeding through here and transcribing things. So
Even though
It's
the
a
whole
roadblock.
answer was like just this huge blob of text, that's all essentially.
Well, for part A, it just says to draw the map.
Understood.
Okay. And then for part B, we're going through different genotypes. And so, do you want to go through part B?
Don't think I actually wrote it out.
Oh.
So the TRP gene encodes the repressor TRPO, is the operator TRPA, encodes the enzyme tryptophan synthetase, a TRPR square, or a capital R square repressor cannot bind tryptophan, a TRPO square operator cannot be bound by the repressor,
Right.
and the enzyme encoded by a TRPA square mutant gene is completely inactive. Do you expect to find active tryptophan synthetase in each of the following mutant strains when the cells are grown in the presence of tryptophan? In the absence?
Oh, sorry, trip R minus. I got that wrong. Cannot bind tryptophan. Oh, cannot be bound by the repressor and trip A minus. Synthetase. Good. So, do we expect to find tryptophan synthetase in each of the following mutant strains when the presence or absence of tryptophan? Okay, so in our first situation, let's do this over. Minus trip and plus trip. Okay, so our wild type situation is R plus O plus A plus. Okay. So in the absence of tryptophan, do we expect to be making trip A?
Yes.
with tryptophan?
No.
No? Okay. That's because tryptophan is going to bind to the trip repressor, and that's going to bind to trip O, and that's going to suppress transcription of trip A. All right. What's our next genotype? R minus O plus A plus.
R minus O plus A plus, R plus O plus A minus,
Sorry. R plus
O plus A
R
minus.
plus O plus A minus. All right. Can we make any tryptophan synthetase from here?
No.
All right, so that's not good for us. We could make it here.
But the promoter's not working.
What's that?
But the promoter's not working.
Well, that's a trip R gene. It's not a
Oh.
promoter.
Oh,
So that's
okay.
an R, not an E.
Oh,
Okay, so this isn't going to be able to bind
Mhm.
to trip O. However, this trip repressor can be made at this locus, and then it can come over here and bind to trip O. And so this is going to behave like wild type.
Yes.
Okay, so it's going to look the same as wild type. Okay. And then what's our last case?
So R is the repressor.
I'm sorry?
R is the.
Yep, R is trip R, that's the repressor, and so that's what binds to the operator and shuts down transcription if tryptophan is present.
Okay.
So that's similar to LAC-I.
Like R, yeah.
Okay. Again, we have these interactions between the effector molecules and the repressor. And our last one, R plus, O minus, A plus.
And then R plus O plus A minus.
Okay. So once again, we're not going to make any
Mhm.
tryptophan synthetase here. We can make the normal trip repressor here and here. And so what we're interested in is what's going to happen at this site. Okay. So if trip O minus cannot be bound by the repressor, what's going to happen in the absence of tryptophan? Will we have transcription of this gene? Yes. Yes. Yes, we will. So we're no longer able to repress it. So if trip R can't bind here, okay, we're just going to have transcription occur.
Okay.
So yes, in the absence of tryptophan, transcription occurs. In the presence of tryptophan,
Okay.
it occurs.
So this type of product is the same as the one you showed in class, right?
As the
The IS
Works.
and
so the trip repressor system, this trip operon system is similar in concept to the LAC operon. It's just a little bit different in terms of how the effector is regulating the operon.
this one does not have the I part to it.
No. So what was LAC-I? Like
Like I was for the inhibitor.
yeah. So we refer to that as a repressor protein. And so so LAC-I was the repressor protein. In this case, trip R is the repressor protein. So they serve similar functions in terms of shutting down transcription, but in different names. Okay. Any other questions? Yeah.
I have a question for the part where you did the analogy for the Moore's law.
Or did I'm sorry?
The analogy for the Moore's law, like one worker is sick and
Oh yeah, sure.
cover.
Sure.
Yeah, I don't understand
Put it
some,
on the side,
well,
put this right.
some of the pick that we did in class.
Okay.
Is it on the, is it on lecture 13? The most recent one?
Yeah, Friday one.
Lecture
That's not gonna be
13
on the-
is not on the exam, right?
Yeah.
That's right.
Uh-huh.
It's in the next exam.
Oh.
So we're gonna
Yay.
Also, quick question. I just noticed this grading. Is the final going to be like just another midterm?
Yeah.
Okay.
It's not cumulative.
Yeah, I just noticed that. So.
Unless you guys want us to, yeah, do that.
No,
Yeah,
no.
yeah.
Make us stress more now.
I'm sorry?
Just make us stress more now.
Make you stress more now. Yeah. I figure there's a lot of material. I think comprehensive final would be challenging. I mean, on the one hand, it's like it's really good to have a comprehensive final because there are, I think, some concepts that people didn't learn so well the first time around, say like the last midterm, and so it would be good to revisit those to encourage you to learn those better. We're going to be talking about a lot of things in the third section of the course, so I just want to make sure that we have enough opportunity to go over that. Yeah.
So during mismatch repair,
Mm.
I'm not sure which protein looks for a methylated DNA strand to determine which one is older. It just assumes that the older methylated DNA strand is correct. That's the assumption that it makes, and it repairs mismatches.
Yeah, so that's correct. So it's using the old strand as the template.
Okay. Checking.
Mhm.
How does it know? But it doesn't. It just makes assumptions.
Well, I mean, so if you're incorporating errors during DNA replication, then that works well.
But if it's errors such as the oxidative stress from, I think it was binding to something which is basically added.
Yeah.
then
So
that-
yes, so it can have problems in other situations. But is mismatch repair called in primarily during DNA replication, or is that? I think that could be a problem.
It doesn't say it's during, oh, it has replication order, so yeah, it seems
Yeah,
like.
replication.
In that example, yeah. So that's when it works well, because once the strands are older strands, so there will be methylation going on at that GATC site, and then it's not clear which strand is the correct strand any longer. And so you're right, that makes it easier to incorporate mutations.
Chapter 8, problem
Do
6.
question 39 of chapter 9.
39 on chapter 9.
Yep. The one that we did. Or 8.
Oh my gosh, that one was so frustrating.
Which one is this?
I was
Chapter
so.
9.
You spent 2 hours on that problem.
We didn't spend 2 hours here.
On which one? 39?
That
Yeah.
was an assignment,
Okay.
right?
I mean, we spent 2 hours figuring.
2 hours on number
We
39?
did not spend 2 hours using saturating. We
Okay.
spent like maybe 30 minutes talking through the answer.
Okay.
Maybe.
Okay.
Generous.
All right. Number 39, chapter 9.
Yeah.
Yes? Okay. So we're dealing with a single nucleotide addition and single nucleotide deletion, approximately 15 bases apart in DNA, causing a protein sequence change. All right. So let's go through this here. So the question is, how good are you at navigating the periodic table? Or not the periodic table, the codon. Periodic table is also very important, just not,
Just slightly, you know?
Do we have to memorize all the amino acids?
No, he's gonna give us the codon table, much like he gave us the.
Oh, you haven't memorized them already?
Yeah, I did.
Oh.
Yeah, I did.
Are you talking, which memorization are you thinking of, the codon table or like phenolphthalein and serine?
The codon table.
Oh.
Oh, no, this
I'm
one
not
is
used to
useful.
it.
If you memorize the codon table, I would tell you that you wasted your time.
Because you know when you need it.
Okay. So this is before mutations, and this is after. And we're trying to deduce what the sequence was that encoded this, and then what the sequence is encoding this after these mutations. So we know that we have an insertion and a deletion in here somewhere,
15 bases.
approximately 15 bases apart. Approximately. Okay, so let's see what we can do. Everybody get out your table? Now, so one important thing, let's start off by saying, where do we think that these mutations occur?
What do you mean by that?
I'm sorry?
Not after the
Where?
first one? After the first
Wait.
one, yes.
So you think here? Okay. So why do you think that there might be, hey, why do you think there might be a insertion or deletion here?
Just a
That's
change.
that's the first one that changed.
Great. Okay. So when we have an insertion or a deletion, if it's not in a unit of 3, that's going to lead to a frame shift, right? And so when there's a frame shift, we expect there to see lots of changes in amino acids, because now we're now translating a different frame. So yeah, that's a great point to pick out where there's likely to be a mutation. Where do we think the other mutation might be?
15 base pair.
Between alanine and valine.
Between alanine and valine?
Methionine and alanine.
How about, yeah, right here. Okay, so what has happened at this position? So here, we had a frame shift. Now we're out of frame, and we don't know if that's because we deleted a base or we inserted a base. We just know that we're out of frame, and we're getting different amino acids here. What's happening at this point?
Back
It's
to
back
the
again.
Yeah,
original.
put it back, all right? So there's probably a mutation here, and a mutation here, and you know what? Looking at this, 1, 2, 3, 4, 5,
15,
okay,
15.
15 base pairs apart. So let's go through and deduce. So the first thing that we should do, I think, is figure out what the possible codons are for this region. What is helpful for you to know is which amino acids have more than 4 codons. So which ones have more than 4 codons? Serine. Yep. So serine has 6 codons. Who else?
Arginine.
Arginine. Yep. Good. Anything else?
Leucine.
Leucine. Very good. Is that all of them, I think?
Yeah.
It's a pain to scan the table. So if you know those 3 ahead of time, then you'll know that you might have to consider extra sequences there. That you might have missed something if you only looked at one spot in the table, is my point. Okay, so we'll watch out for serine, arginine, and leucine. They're tricksters. All right, so phenylalanine can be U, and then let's say U or C. Serine can be a UCN or a AG, U or C. Proline is CCN. Arginine,
CGN.
CGN or
AGA or AGG.
AG or G. Leucine,
CUN.
CUM.
UUA, UUG.
Sorry, what was that?
UUA, UUG.
UUA or G. Scourgine is AA.
U.
Disgusting. So let's look at this first situation. We have a serine turning into a valine. So what are the possible valine codons? It is going to be G, U, N. So if we look at the possibilities here of how we could shift from a serine to a valine, what could that be? So in one case, let's say that we delete a base here. Would that work? So if we,
Yeah,
let's
you
say that we
deleted
delete,
A.
well, we delete that. What's that?
We deleted A.
Yeah, so if we delete this, then we could end up with a shift here to G, U, C. That would work. If we added a base,
You could insert G. You could insert G just before the area to get to.
that's
G,
correct.
U,
Yeah, so we
C.
could have added a base here and done a G, U, C. So there's two possibilities there. And so if we added the base and it went G, U, C, then we would have to go NCC for NCC. What's histidine going to be? Histidine is going to be CAU or C. And so say we added a G there, then we can't get that histidine, right? And so what we can do is so that option's out. All right, so say we shift like this, and then we shift here, and shift here, shift here, and then something's going to happen. All right. So we've now deleted this base, which means that our new codon is going to be like this, this, and this, until we get to that other insertion. All right, so of these possibilities, this one doesn't work, but GUC does work. All right, so let's write that in here, GUC. And we'll just get rid of this guy. All right, so the next one is going to be C something, C or A. And so for histidine, which one works? The C, A, C. All right, so now we know that this is CAC. All right, now we're going from arginine to alanine, and let's see, alanine is GCN. Okay, so which of these could work? We can either be GNC or GAU or GGU. Okay, so we're going to do the GNC, and specifically, it's going to have to end in a C. Okay, so this one's out. So GNC, and so in this case the N is a C, and this N is a C, so it's going to be GCC. Okay. All right, leucine to leucine. Okay, we have two possibilities. It can be CUN, or it can be. Oops.
Start with U.
Start with U. Thank you. UA or UG. What are we doing here?
Because you ended with C, and then you go UN, and then to the next one A, right? For the codon for the C.
Did I write something down incorrectly?
So for alanine, you did GNC or GCC.
That's right.
So you cut in. So for
Yep.
leucine underneath, wouldn't that be UNA?
That's right. So that's right. We're going to be going to that. So we're going to go with UA for this codon. Because we can't start with a C. We know that the first base is going to be a U. So let's get rid of this possibility. So it's either going to be UUA or UUG. So the only option that works is if we this one right here. Okay, so it's UUA. All right. Methione's great, so that's going to be an AUG. There's no getting around that. There's only one codon for methionine. All right, and now we expect something to happen here so that we can get back to doing GCN, GUN, AA, A or G. Okay. So how can we do that? Because here was our start codon. Insert a G. We insert a G right here. All right. And now we're back to normal. Okay, so now that we have deduced what this sequence is, we should be able to say what the sequence of the top strand is, because the only thing that we've done here is we did a deletion here, and we did an insertion here of a G. or serine originally?
AGU.
What's that?
AGU.
There's 6 possibilities for serine.
But for the GU is only AGU.
Was it UCC? Was the original?
AGU.
AGU. Right here. Thank you. Okay, great. All right. So it's kind of a pain, but it's doable if you pay attention to which ones have the 6 codons and work. So the nice thing about this is because we had this frame shift, all of a sudden all of these ambiguous bases that were present in the 3rd position can be resolved because we've now shifted them to the 2nd position, and that's much more restrictive. And so we're able to deduce most of this sequence in here, just not at the ends. All right.
But we need to know the very first 3, or would it be okay to put the first 3 as well insert if we don't know exactly?
Oh
the
yeah, I mean,
numbers?
we don't have enough information to know what goes here, so you would have to list it as something ambiguous like this. There are codes for, if it was U or C, there's a code for that. There are other letters.
Would that be fine on the exams writing like U over C?
Yeah, that's fine. Okay, you good? Yes. The Ames test? Sure. Is there something specific you want to know about the Ames test, or?
The first question I have is it just a yay or nay, is this mutagenics, or does it also discuss how mutagenic it is?
I think I would expect there to be a degree of mutagenicity. Yeah.
So then how, what is it test, like, so you set up everything on the graph, but what do you set up on this graph, how do you get error?
Okay. What is it testing? So there was a graph that we showed. Is that the one that you're referring to?
Yeah.
Or is- Okay. Yeah.
Yeah.
All right. So in this graph, we're using the Ames test to determine, well, whether or not a particular compound was mutagenic. And so remember that we were dealing with 3 different mutant strains, and they had different types of mutations. So one of them had a insertion or a deletion. Which one was which?
It was, TA100 is a base substitution.
Base substitution. Okay.
1535 is an indel, and 1538 is also an indel.
Okay. Okay. So in the problem that we just did, there were 2 mutations, and we were able to, one mutation introduced a frame shift, but then another mutation restored it back to the normal frame. So the way that you're going to fix an indel is through another indel. The way that you're going to fix a base substitution is through a base substitution. So you're not going to be able to fix a base substitution with a frame shift. Does that make sense? So when we're talking about reverting a mutation, restoring the gene back to normal function, what we're asking is what sort of mutagen is this? So one, does it cause reversion of those original mutants? And if so, does it specifically cause reversion of one type of mutant? And that can give us clues about the types of mutations that it's causing. So we're adding a mutation to fix a mutation. So when we look at this graph, we have an increasing dose of this compound called aflatoxin. And we're quantifying the number of revertant colonies per plate. So remember that you're taking your mutant strain of bacteria, you're mixing it up with the aflatoxin and probably some liver enzymes that have processed that aflatoxin. And then you plate them out, and you're looking at their ability to survive in this particular, they're oxytrace, right, for, I don't remember what. Let's say they're oxytrace for histidine.
Histidine oxytroph.
Histidine oxytrace, right? So what does that mean if it's a histidine oxytroph?
It needs to have histidine in the medium in order to live because it cannot create histidine.
That's right. Okay, so it's got a defect in a histidine biosynthesis gene, and so it can't make its own. You supply it histidine, it will grow fine. And okay, so what we're asking is, once we do this incubation, and we put this on a plate that is lacking histidine, the question is, how many colonies do we get? So the more colonies we get, the more revertants we have. And remember that in order for us to get these revertants, we have to be introducing new mutations. Right? So this broken histidine biosynthesis gene, the way that we're going to fix it is through additional mutations. So when we see this increase in the number of revertant colonies as we increase the aflatoxin dose, what is that telling us? That's telling us, well, that yes, this is able to cause mutations. And we can see that clearly happening here. It's happening in dose-dependent manner. So more aflatoxin, more mutations. However, it's not affecting these two strains, which have the indel mutation. It's only affecting this strain that has the base substitution. So if we were to make a hypothesis about what types of mutations aflatoxin is introducing, what would you suggest that might be? Base substitution. That's right. Okay. We're fighting fire with fire, so to speak. So does that answer your question? Okay, great. What's that?
What number is it? 20, chapter what?
Back on the lockout then?
The same chapter.
Blackout one. Would it be transcription?
All right. So this is number 20 from which chapter?
11.
11.
Okay, cool. Just for posterity's sake.
Yeah.
20.
Problem number 20 from chapter 11.
Eleven.
11.
Number
20.
20. All right. So we have 7 new mutant strains that lack the products of all 3 structural genes. Good. And here they're referring to Z, Y, and A, I believe. Okay. I suspect some of these are lacked by S mutations and others.
The downstream of permease, like operator has a more.
But does it have a function?
This is like
Or transfer?
it's like personalized. Yeah, I remember that.
Okay, 11.
All right. So remind me, what's wrong with lac P minus?
Like the promoter, no promoter. The promoter doesn't work.
Promoter doesn't work. So what does that mean?
Like no transcription at all.
Okay, so RNA polymerase isn't able to bind. We have no transcription. Doesn't matter if lactose is present or not.
The promoter region is where the polymerase binds, correct?
Mm-hmm.
Okay.
So not a LAC-O mutation, not in the operator,
Okay.
LAC-P. All right, and then the IS mutation, what is that?
Wouldn't be
Not a
able
lactose
to.
insensitive?
Okay, so what's the net effect?
Wouldn't be able to bind to the effector, like binds to the operator all the time.
That's correct. Okay, so it binds to operator. All the time. Equals no transcription. So, both of these mutations result in a lack of transcription from the lac operand. How can we distinguish between mutants that are lac IS versus lac P minus mutants?
One is cis and the other is trans.
I'm sorry?
One acting cis and the other acting trans.
That is the key, right? So which of these is going to act in cis?
P minus two.
Okay, so this acts in cis. And this acts in trans.
Trans, yeah.
Okay, that is the most important distinction between these two. All right, so let's see if we can come up with an experiment here to distinguish this. Any ideas?
Can we do two experiments or do we do?
Yeah, you can do whatever you want. Yeah, sure.
We can do a reaction where we have IS P positive with the Y positive P positive.
Do you want to write it up? Sorry, I'm hard of hearing, so this will just say this all over. Here, try this. The curse of the whiteboard markers.
Need to borrow it.
Yes! With the bank shot.
Bike shot.
I always miss Mama and SI kiddos make fun of me.
I
Bike
have
shot.
them.
You guys should be thinking about this too.
I am.
You're thinking?
Yeah.
What are you thinking?
Creating experiments.
Okay. Let's come up with an experiment here. All right, so let's walk through this example. What will happen here?
This would be able to because this acting trans, it's all in one copy.
Good. So that IS will bind here or here and shut things down. Okay.
Wow.
No, you
This,
better wild type for
because
both.
this is.
Do we have to consider presence of the operator as well?
Yep. So, well, in this problem, we don't necessarily need to call in the operator, but you could. So, let's see.
Because when you
I
have a
guess
mutation in the
it'd be
operator,
better
it's gotta be
we
different.
include the operator because
The-
this binds to the operator.
Mhm.
I mean, I'm just gonna.
Yeah, I mean, so the
That's-
operator needs to be present, of course. I'm just trying to think, so we have mystery mutants, right? And
Do we know anything about the genome of the mutant? So do we know if it's simply one area that's affected, or could there be multiple areas that's affected, such as IS in tandem with Z minus, Y minus?
I think we'll just assume the simple situation, that there's a single mutation.
Okay.
All right. So we have mystery strain, right? And so what if we could introduce one of these F prime plasmids into the mystery strain? Right. So what we don't know is whether our mystery strain has this lac IS or this lac P mutant situation. And so if we give it, by the way, it's very brave. I think that the trick that we're going to have here is we want to ultimately introduce this guy in somewhere, and so we need to figure out where that's going to fit in with these tests.
So this has to be working with that like that too.
So you are, I think what you let's see.
We are trying to
I think
create
if
the
we-
experiments to,
We
Yeah, to
say
figure
like
out what the
this
function is, right?
yeah, to,
makes it
And we're only
look
up
a lot
to
like that,
7
not even
genomes.
with the IS. Right?
Couldn't you make it
And
at
then
the
the other
time
difference
cost
with the
then?
this
IS.
is like,
and
it's
then-
working right now.
Able to find.
Yeah, this is a-
Do we know?
This is a good question.
Yeah, no, we're talking about the induced and non-induced thing. So if it.
Oh, okay. So plus
I think what I would
in the,
go with is we're going to introduce a
for here.
normal, we're going to introduce
No,
an
it's
F
gonna
prime
be wild
plasmid,
type, right?
and it's going to have
Yeah, but then there's still P minus here.
Doesn't matter for this one, it's gonna be wild type or.
Oh,
Wild
P
type or
minus.
yeah.
Could we introduce an plasmid with all of the genes as the wild type, like I plus?
Sure,
O
if you
plus,
understand the
blah, blah.
dominance between these. Yeah.
And then, because if it was gonna be P minus, you would still see Z and Y because it would only act as cis, and so one of the two genes, or one of the two operons would still be producing Z and Y. Whereas if you saw IS, considering that acts in trans and a very dominant phenotype, you wouldn't see any Z or Y.
Yeah, I agree. So you could put it, so you're saying put in the wild type on the F prime plasmid, and you're saying that if we have the lac IS on this other location, it's going to be binding on here and shutting this down. We see nothing all the time. And you're saying that if it's a lac P type of mutation, then
That would still produce, that normally
This is
produces
going to keep
P
doing
when
its
the
thing.
X is cis.
yeah, I think that works well. I'm trying to think if there is a way that we can do it to be absolutely sure that it was a lac P minus mutation and not something else, but I
Like
think
an O minus mutation?
ELOC or
C, sorry.
Let's see,
I think that this test that you're proposing is probably the most straightforward way to do
what
it.
is it again?
Hmm?
What is it again that she proposed?
Her proposal was to put in just a wild type copy of that lac operon. And because lac P minus is going to be acting in cis, that means that if there's a lac P mutation at the locus that's in the genome of this mutant,
Nothing's gonna.
well, it can't affect what's going on here. So you're going to see your normal responses to the presence of lactose. So lactose will be able to induce expression minus plus minus plus pattern.
Are you
If-
saying putting
Yeah.
P in the same locus, like the P minus?
No. I'm saying, okay, so here we have our normal P plus, I plus, everyth's wild type,
Mhm.
Okay.
okay? And the point is that if in our mutant, we have two options. One is that it is IS, P plus, O plus. And the other is that it is I plus, P minus, plus, I plus. Okay? So if it's this situation, I'm sorry, so if it's this situation, this guy can travel here and shut this down all the time,
Yeah.
right?
Mm-hmm.
If it's this situation, well, it doesn't matter if it's this or this. This one is going to not be able to go here. Oh, I'm sorry.
No.
I'm sorry.
It has nothing to do with that.
Thinking. It will still bind here, right? But the point is that there will be this defect, and so we're just never going to see any transcription responses at all. So here we should be able to. So how do we discriminate this? I just confused myself.
I thought the problem.
Because the P plus on the F prime plasma would still be able to, if we assume that it was I plus, not I S or anything else, since P acts as this, the effector would still, you'd still have the same normal response to the effector because the F prime
Yeah,
is
yeah.
wild type and P only acts as cis.
Yep, so we get expression or we get no expression. Yeah, right?
What,
Well,
how
there's
can you
no
get
looking.
expression if we?
Sorry. Okay, let's redo this. Let's redo this, okay?
Right?
Yeah.
Here, we'll write it up. Okay. IS, P plus, O plus, Z plus, Y plus, or we have I plus, P minus, O plus, Z plus, Y plus, and then we also have our F prime. We have I plus. Or we could do, if you want to get rid of this altogether, you could be I minus. P plus, O plus, Z plus, Y plus. Might be the cleanest to do this with I minus. Okay, so if we have these two guys together,
Mm.
Mhm.
okay? So
Oh, transfer, like no expression. Okay, got it.
this guy
This
is going to shut down
I have.
expression,
What's my problem?
right? So we see no expression, if that's the
Okay.
case. If we have this situation, I gotta write this plus. This, why do I keep doing this backwards? We have this situation, and we have a normal black eye, which can bind onto here, and it can regulate the black operon normally.
Mhm.
I like this.
Oh, yeah. If you-
Basically, what we're saying is it's either going to be wild type black eye or this black eye S version.
Mhm.
We're distinguishing between those two possibilities. And because this black eye can move around and regulate our locus here, we're basically going to look at whether or not we get no expression or expression coming off of here. We're never going to have any expression coming off of these.
So that expression is only you're talking about that locus.
Yeah,
Wild type.
yes, we're ending up looking at this locus. So in this case, you agree, you can't get any expression here,
Mm.
okay? In this case, the problem is that black eye is going to suppress it here, and it can also suppress it here.
Mhm.
So I would use the I minus here just so that you negate any effects of this. We want to have whatever this black eye is to come in.
What if there's other plus prime?
Well, we can say what we're putting in here.
What's that?
We can decide what genotype we're going to put on the F prime plasmid.
No, like what happens if
So black eye can't bind to the operon, so it's just going to kind of Did you guys have any other questions?
I think it's funny though.
So what was that?
I did a great job explaining that one.
So will this midterm probably be towards like the last one, or?
Review.
Just- Okay.
Let's see what happens. So,
Yeah.
up?
Try
Oh,
something.
just real quick.
would it make a difference?
Wait, just real quick. So for okay, so replicative equals copy and
Copy.
paste equals retrotranspose. And then conservative is
DNA.
equals DNA equals cut
Cut
and
and
paste.
paste.
So conservative, think about you're conserving the number, so there's one transposon, there's still one transposon.
Yeah,
So
so that's why there's not a lot of not
Like cut and paste.
multiplying or whatever.
Yeah, it's just cutting itself out and inserting somewhere else. And replicative, well, you're replicating it, yeah.
I do have a question on that, say problem 1120. It gave an example in the answer, like I know it's just an example on how
Which
to
one?
tackle it. Chapter 11, problem 20.
Oh,
What
okay.
they gave was an example. What does it mean by constitutive?
What does it mean by constitutive?
Yeah. it was a little confusing.
Constitutive, that word means, basically it's on all the time.
Oh,
The
okay.
number.
Alright, and then could you go for problem, or chapter 8, problem 6?
Which one?
Chapter 8, problem 6, the diagram kind of confused me.
Where'd you see it?
No.
This is chapter 11, number 20.
Constitutive. Like it's all.
Chapter 8,
I know. Phil, what were you?
problem 6?
Never mind.
Chapter 8, problem 6.
Okay, so this is-
It's referring to the figure, and
Mhm.
when I looked at the figure, it just confused me, honestly.
Okay, let's look at it.
Number
So,
six.
we're looking at figure 815. And we're talking about a G to A mutation in the first G residue of the intron.
You have to read. What class?
I have two of those church. And then we'll be back. Well, I won't be back here, but.
What figure is that? Oh.
815.
Okay, so do you see those numbers that are in this figure, these numbers underneath?
It says frequency of occurrence, and then it also gives the number of bases, so
Like the percentage.
So what these numbers are referring to is the percentage of how well they're conserved. So if you take a bunch of introns and you line them up, you do this alignment, you're gonna find that at the beginning of the intron, it's pretty much always GU, and at the end of the intron, it's pretty much always AG. And as we learned later in that chapter, those guys come into play during splicing. So if you were to mutate that base to an A, what is likely to occur? Let's go back and look at our splicing. So if you remember if you look at figure 816, okay, so figure 816 goes through the mechanism of splicing. And so that G at this position, that is where that first point of attack is, right?
but it's not gonna actually go through that snipping process.
Right. So if we have this
Mutation.
mutation here,
Mutation.
we can't have that attack occurring. We're not going to be able to splice out the intron.
Okay.
So what will be the effect on splicing? We're going to have a defect. Okay.
Yeah, it's not gonna slice it, so it's just gonna pass through and affect the MR.
Yeah. So either this won't be spliced out at all, or maybe there happens to be like another AU somewhere that works
Makes it
as a
smaller.
splice site, but it's unclear. The point is that it's not going to splice correctly.
Okay.
So lysosome looks on those two like nucleotides in order to initiate splicing.
Yeah, so if you look at figure 816, you can go through the mechanisms of
Mhm.
how that splicing occurs. So there's the internal
A.
A here, which is going to make the attack on
U2.
that, right? And so that forms that lariat. Okay. All right, we good?
I thought the A right there was the turning point, the G, the A, no, the hydroxy group from the A attacks what used to be another A, and it's now a G.
So the initial one is the hydroxy group from the A attacks.
Mhm.
Attack is base.
The G.
Oh, it was,
Yeah.
okay.
But it's a
We
point
do A-
mutation.
Okay.
There's a point mutation over there, which is why splicing won't occur over there or might not occur at all downstream.
Initially, this is going to attack here and form the
Mhm.
lariat. There's typically going to be an AG here, and this is going to wrap around that. And then whole thing goes. All right. Any other questions, or we good?
So I'm assuming on the material will be given the codon table.
Yeah.
Anything else we're given?
Wobble.
I'm sorry?
Wobble.
Yeah, I can put that wobble table on there, that's fine.
Oh,
New.
that's right.
The wall table.
Wobble table. Wobble table.
Great. All right, thanks, guys.
Anti-codon and codon.